Hello everyone, welcome to MedDevice. In this video, we're gonna be looking at a few previous year questions from the subject of biochemistry. So let's start. So the first question is, which of the following substrates of the Krebs cycle is reduced by excess ammonia? Is it malate, fumarate, pyruvate or alpha ketoglutarate? So here the correct answer is alpha ketoglutarate. I'm not going to tell you the whole Krebs cycle now because it will be a very long video. I'll make a separate video on that. But for now, all you have to know is that there's a substrate in Krebs cycle by the name of alpha ketoglutarate. Now this alpha ketoglutarate reacts with ammonia to form glutamate. This happens with the help of glutamate dehydrogenase. Now again, this glutamate reacts with ammonia to form glutamine with the enzyme glutamine synthase. So first you have alpha ketoglutarate, then glutamate and then glutamine. Each step ammonia is added. Now the question said that what will happen if ammonia is increased? So in hyperammonemia, the excess ammonia reacts with alpha ketoglutarate to form glutamate and then glutamine. So there will be an increase in glutamine and a decrease in alpha ketoglutarate because the reaction is going to move in the forward direction. Now since the alpha ketoglutarate is used up, the TCA or the Krebs cycle will not be able to work and so the energy formation will be not there. So there will be a decrease in ATP which can result in loss of consciousness, coma or even convulsions. So coming back to our question, our correct answer is option D. Now, what deficiency results in increased glutamine levels in the bloodstream? Is it fumarate, ornithine transcarbamylase, arginase, or arginine? So for this, you should know the urea cycle. So let's quickly review that here. So here you have the mitochondria, and the first step of urea cycle is where ammonia reacts with carbon dioxide. This forms carbamoyl phosphate with the help of carbamoyl phosphate synthase 1 or CPS1 for short. Now we have ornithine which reacts with carbamoyl phosphate to form citrulline. This happens with the help of ornithine transcarbamylase. Citrulline reacts with aspartate to form arginosuccinate, and the name of the enzyme is easy to remember, arginosuccinate synthase. Now this arginosuccinate breaks and gives out arginine and fumarate, and arginine finally forms back ornithine, giving out urea. This is done with the help of arginase. Now the question was, which enzyme deficiency here can result in increased glutamine levels? So the correct option is ornithine transcarbamylase. Now why is that the case? So let's say that this enzyme is not working, the ammonia levels are going to increase because the reaction is not going to move in the forward direction. Now coming back to our reaction which we just learned, you can easily understand now that if there is an increase in ammonia, the reaction here is going to move in the forward direction, meaning there will be formation of glutamine. This is why the deficiency of ornithine transcarbamylase can lead to an increase in glutamine levels. So coming back to our question, our correct option is option B. Now, which of the amino acids listed is required for the conversion of norepinephrine to epinephrine? Is it tyrosine, phenylalanine, methionine, or glycine? So let's quickly see the formation of norepinephrine and epinephrine from the start. So first of all, we have phenylalanine, which is a very important amino acid that you should know about. This, with the help of phenylalanine hydroxylase, gives L-tyrosine. L-tyrosine, with the help of another hydroxylase, gives us L-dopa, which gives dopamine with the help of decarboxylase. Now this dopamine gives norepinephrine with the help of an hydroxylase. So the correct sequence of the enzymes is hydroxylase, hydroxylase, decarboxylase, and then finally hydroxylase. Now this reaction of norepinephrine to epinephrine is what was being asked. So with the help of phenylethanolamine, N-methyltransferase, and SAM, which is S-adenosylmethionine, we get epinephrine. So here if you will focus on SAM, you can see that methionine is very important for this reaction to happen. So methionine is the answer which we were looking for. It is the amino acid which is required for the conversion of norepinephrine to epinephrine. So our correct option is option C. Now, what amino acid is used in the synthesis of nitric oxide? Is it tyrosine, glycine, arginine, or tryptophan? So the correct option here is arginine. So arginine reacts with oxygen to form nitric oxide and citrulline with the help of nitric oxide synthase. Now, what are the other important things that you should know about? So, nitric oxide causes vasodilation, which helps in regulation of blood flow and the blood pressure. It also helps in penile erection. Nitric oxide has a bactericidal effect. It is also a neurotransmitter and it is an inhibitor of platelet aggregation and adhesion. These are important one-liners that you should know. So coming back to our question, our correct option is option C. Which enzyme would be affected in Menkes disease? Is it lysyl hydroxylase, lysyl oxidase, propyl oxidase, or propyl hydroxylase? So what exactly is Menkes disease? So it is also known as kinky hair syndrome. In this disease, there is a mutation in the ATP7A gene. It is an X-linked recessive condition and there is a defect in the copper binding P-type ATPase. So due to this defect, the copper is not mobilized from the intestine, and copper is a very important cofactor for an enzyme by the name of lysyl oxidase. So if you don't have copper, you don't have this enzyme, and this enzyme helps in catalyzing the cross-linking of collagen and elastin fibers. So coming back to our question, our correct option is option B or lysyl oxidase. This is defective in Menkes disease. Now, what is true regarding mitochondrial genome, maternal inheritance, low rate of mutation, single-stranded, or paternal inheritance? So, what is the important stuff that you should know about mitochondrial genome? So, first of all, it is double-stranded and circular. Secondly, it has maternal inheritance because only the maternal mitochondria are passed to the offspring. There is high rate of mutation seen. Finally, another important point is heteroplasmy. So heteroplasmy is when there is more than one type of mitochondrial DNA in an individual, which determines the severity of mitochondrial disease. So coming back to our question, low rate of mutation is false, single stranded is false, and paternal inheritance is not seen but maternal inheritance is seen. So that is our correct answer. Now final question, NADPH finds its application in fatty acid synthesis, ketone synthesis, glycolysis, or gluconeogenesis. So NADPH has a lot of uses. Let us look at some of them. So first of all, it is involved in reductive biosynthetic pathways. So it is used in the formation of fatty acids, steroids, and some amino acids. Then it is also involved in the regeneration of glutathione. Third, it is a cofactor for microsomal cytochrome P450 enzyme system. Fourth, it is also used to protect against a few bacteria and it has a few special functions. So we already saw that it is responsible for regenerating the glutathione, and this glutathione is important for the integrity of the RBC membrane. Finally, it is also important for keeping the iron in Fe2 form. You must be knowing that if hemoglobin has Fe3 plus form of iron, it leads to the formation of methemoglobin. Now this is a bad thing and we don't want it. We want the iron to be in Fe2 plus state. So here also NADPH comes in play. So coming back to our question, our correct option here is option A since it is involved in the fatty acid synthesis. That's it for this video. If you have any doubts, so please leave them in the comments or feel free to message on Instagram anytime. The link to my Instagram is in the description. Thank you.